So we have to speak English now.
Yes.
T'as pas ouvert la... Ah non.
Thank you very much, Anne, for the very kind introduction. I just wanted to double check whether everyone can hear me fine. So I would just like to perhaps continue from Anne's introduction to say that actually this talk here in Montpellier marks a really wonderful story of scientific interaction that spans actually 20 years. It's a European experience. Because I actually came over to Montpellier for the first time 20 years ago as an Erasmus student, and I worked with Nicolas Bierne in ISEM at the time. And that's when I was given a book of Jacques Blondel on evolutionary ecology to read, and this is when the adventure started. And I had the great privilege to collaborate and befriend many CNRS from Montpellier researchers over time. So now I'm going to share my screen and present you the talk that, okay. All right. So today I will be, oh, and I'll just start my timer as well. So today I'll be talking about life on our doorstep, and as most of us live in urban areas, life on our doorstep is really about urban ecology and evolution. So there has been much focus over the decades to really study ecology and evolution in a natural setting and to study evolutionary dynamics in a natural setting, specifically because this is where the environment fluctuates, and this is where we can best understand patterns of selection and response to selection over time. And it is true that Charles Darwin, when he himself formulated an understanding of evolution, he was actually traveling around the world and observing natural phenomena in natural habitats to grasp this idea of how we came to be. However, what is also interesting is that humans, like ants, are ecosystem engineers. And what's fascinating in this comparison is that ants evolved around 140 million years ago. And within that time frame, there's around 10,000 species of other insects that evolved to take advantage of the resources that ants created as ecosystem engineers. While we, in contrast, have been building cities only for the past 200 years. So since our original times, as hunter-gatherers, we have transformed the environment with increasing speed, and we have transformed the ecosystem that we live in, from savannas to agricultural lands to cities, finally, where we have actually gathered an increasing amount of energy into the ecosystem. And so we are thereby getting to the flagship construct of our times, and these are cities. So urbanization is a growing phenomenon. There is more and more people living in cities. At the moment, we have probably crossed the threshold of more than half of the world's humanity living in urban areas. And if we can, and it's not a phenomenon that is only specific to one continent. Cities are growing worldwide. And so you can see here that essentially circles are scaled in proportion to urban population size. And what you can see is that more and more people worldwide live in cities, and consequently, the space that is occupied by cities is increasing worldwide as well. And it is estimated that around 3% of the land area is currently occupied by cities. So as it has been phrased by Hutchinson in '65, ecology, we can talk about the ecological theater and the evolutionary play. And Marina Alberti recently has put that in a context of the urban environment, that by changing the ecological theater, cities are simultaneously changing both the actors and the stage, thereby leading the way to a new eco-evolutionary play. So cities are this construct element of the Anthropocene we're living currently. And by just continuing this narrative of a theatrical play, I would like you to present you some data about the Anthropocene and the urban environment, and how by talking about Nestboxes, cities, and parallel cities, we can gain a greater understanding on the evolutionary dynamics that are occurring in the Anthropocene. So First of all, I will start by discussing the natural world. So as I was saying, a lot of knowledge about the natural world that we have gathered is in seemingly natural habitats, such as this picture of Whiteham Woods in Oxfordshire in the United Kingdom. And we would imagine that by looking at nest boxes, at birds in nest boxes, we are capturing some element of the natural environment. However, to what extent, what does it mean natural? Are we actually really capturing natural variation in these forests? So we have to remember that a lot of vertebrates, that most of our natural wildlife essentially originally evolved in natural primeval habitats, but we hardly have any primeval habitats left in the environment. And essentially, all what we're capturing currently in the so-called natural habitat stems from secondary forests, and a lot of information, fantastic information that we have gathered in evolutionary ecology stems from these nest boxes. So I don't think I need to tell much about the amazing data that has been collected across Europe that we have collected from our nest box study sites. And nest boxes are incredibly useful because they're easy to follow. Birds breed readily in them, and it is easy to collect large sample sizes, which are often essential to really have a grasp on the ecological and evolutionary parameters that are essential to understand how organisms respond to a changing environment and how they adapt to their natural environment. And so these pictures are like a typical of what you would see from a evolutionary ecology study of birds. But here I'd like to present you the work of Joanna Sudyka and Irene Dilece that have gone beyond that, and they have asked, well, what is happening really in natural cavities? I mean, does what we can observe in nest boxes actually reflect natural trait variation of birds that originally are breeding in natural cavities? And if we are to look into fieldwork collected, if we're to look into fieldwork by looking at natural cavities of a model study system, such as the gray tit or the blue tit, fieldwork suddenly becomes quite much of a bigger task, because instead of just, for example, checking all the nest boxes in a certain sequence, you actually have to start early in the season and identify the natural cavities, and you need to get to them. And finally, to collect phenotypic or life history traits, you need to extract data that is often quite challenging to get. So you can see here Joanna Sudyka that is trying to extract some nestlings from the natural cavity to be able to weigh it, to collect blood samples, or to ring at them. So what I'd like to show you here is that essentially we took an experimental approach to try and understand whether natural variation in nest boxes is equivalent to that in natural cavities. And so this is the result of work carried out in 2018 and 2019, where essentially we set up, we're essentially in a true urban forest, in an urban protected forest that has remnants of primeval habitat. So with a complex phytosociological structure with a lot of old trees, essentially we have been following breeding events both in natural cavities, and we have also erected an experimental nest box site just across the road, where the environment doesn't vary, and the only thing that differs in the environment is the presence of nest boxes. And what you can see is that the density of breeding events of both blue tits and gray tits is actually highly equivalent when we look at the natural cavity site, which is just here, and when we compare it to the nest box cavity site on the other side of the road. Moreover, if we look at 2019, similarly, the trend is quite similar. So densities don't really differ between natural cavity and nest box sites. What is interesting is that if we look at the microclimate of nest boxes, we actually see that natural cavities have a much more stable environment in terms of temperature and also in terms of humidity, which is the data that I'm not showing. Well, in terms of the nest box, actually the temperature profile of a nest box follows very closely to the temperature profile over 24 hours of the ambient temperature. And if we look at these effects of natural cavities, nest boxes versus natural cavities in terms of life history or fitness, there are actually differences in the reproductive cycle. And so, for example, blue tits in nest boxes lay considerably earlier relative to blue tits in natural cavities. They lay larger clutches and they take longer until fledging. Gray tits, on the other hand, have a shorter onset of incubation and also leave the nest later. So what we have found in our 2-year study is that actually there are some year and species effects. But if we compare this data to data collected by Catherine Purcell in 97, like in a study published in 97, where she compared the breeding ecology of birds nesting in boxes and tree cavities, we also see that essentially there are some parameters that she found that are significantly different when these two types of cavities are compared. And what you can see here is that essentially the profile of breeding of blue tits and gray tits in Europe fits very strongly the patterns that Purcell observed in species in North America. So in some ways, what is possibly concerning here is that lay date and clutch size are key life history traits that are also used in our understanding of evolutionary dynamics in the natural environment. And so to what extent what we are really measuring in nest boxes represents what the large bulk of the population is actually going through, because nest boxes are not present across the entire natural habitat. These are model systems. So summing up this part of the talk, I would like to say that what we found is that there are similar densities of birds in nest boxes and in natural habitats. And in some ways, this is really good news, because this could also be perceived as experimental evidence of the fact that when presented with the possibility, birds prefer to breed in nest boxes, because the densities in both natural cavities and nest boxes was highly similar. There are definitely different temperature and humidity profiles of nest boxes relative to natural cavities. And most importantly, there is definitely a different pattern of life history dynamics in these nest boxes relative to natural cavities. And so the question that I'd like to leave you with is whether estimates of selection and response to selection derived from nest box studies are truly reflective of natural variation occurring in natural cavities where most birds are still breeding. So I don't think it's necessarily a problem if we are talking about, for example, a certain correction factor. So imagining that essentially all birds in nest boxes breed by a constant number of breeding days earlier than those in natural cavities. But the question is to what extent we can really say that, for example, what we have been investigating in longitudinal studies really reflects of what is happening in natural cavities. Perhaps there is a stronger buffering occurring in natural cavities. I think this is a highly controversial topic, and it doesn't make our data interpretation easier, but I will leave you with this because this is certainly something that we have to keep in mind when we are trying to derive specific estimates of fitness in the wild from nest boxes. So I would like to now move on to the next part of my talk, and that is specifically to discuss the city. So 5 years ago, I started a project where the idea was to study gray tits and blue tits in a gradient of urbanization in Warsaw, and to try and study concomitantly the effects of phenotype, genotype, and fitness in a radically altered environment that is linked to urbanization. And to give you a little bit of context regarding how the study started, well, this is Margaret Hamilton, and you can see here her code that allowed her to send people off to the moon. And this is essentially the admin that was necessary to establish a study site in an urban environment. And that stems from the fact that there is a lot of sociocultural particularities depending on the city where you are working that makes field work in an urban environment more or less difficult in Warsaw because of changes in property laws related to nationalization and then restitution of property. It was actually very difficult to get permits for the different study sites that I'll be talking about, but we made it work. So yes, that's definitely not as big a feat as sending people to the moon, but I was still very happy nevertheless that it succeeded. So what are we left with, or what are we starting with? Well, here you can see the city of Warsaw, and the city center. Warsaw is a capital city of Poland with around 2 million people living in the city. And you can see here that essentially we have established 9 study sites across a gradient of urbanization in Poland. And you can see that actually these sites are here set in those kind of dyads, and this is not a coincidence. That's because I tried to essentially identify sites that are close to each other, but that are highly contrasted in terms of the vegetation properties of sites, because that will further on allow for some more experimental work once we have characterized the system. So how do we define the urban space? When you actually look at the studies that have been reviewed thoroughly by Mark Johnson and Jason Munchie-South in their seminal paper from 2017, Evolution of Life in Urban Environment, you actually notice that 3/4 of the studies of urbanization impact on the biology of wildlife use qualitative terms to define the environment. So essentially, mostly what you find in publications is urban and rural. And only 1/4 actually tries to take a quantitative approach to urbanization. And when you look into these quantitative studies of the urban environment, many people would characterize it either with land cover classes, or they would use the percentage of built-up area or impervious surface. And it is true that there are many, many ways you can define the urban environment. And that depending on the biological question that you're asking, some variables may be more relevant than others. And the thing is that you can quantify the urban environment using remotely sensed variables, or you can actually be in the ground and collect data that is specifically relevant, and that you can only collect being on the ground. So here is just a list. And that these different axes of environmental variation are likely to sometimes impact different aspects of the biology of the species you're looking at, but it can include impervious surface area, so concrete and asphalt, and DVI, so the photosynthetic activity of the environment. You can look at tree cover, light pollution, air pollution, distance to roads, sound pollution, or human presence. I would like to focus largely on this index of impervious surface area, so essentially the amount of concrete and asphalt that is in the environment, because it is a highly repeatable measure that you can also easily compare across studies. And it essentially tells you whether there is biological life that is covered, or, sorry, it essentially tells you how much of the environment does not have any biological activity because it is essentially covered in concrete or asphalt. So what my PhD student Michela Corsini has done here is a visualization of the amount of impervious surface in the 516 nest boxes that have been erected in the gradient of urbanization in Warsaw. And you can see that these are our 9 study sites, or actually 8 represented here because one natural forest, I think, isn't presented. And what it shows you is that essentially the urban environment is not like a smooth line of imperviousness from the city center to the natural area, from a natural space. What you see is that the urban environment is a mosaic of heterogeneous environments that have more or less impervious surfaces around you. Intuitively, of course, you know that you have urban parks and you have office areas in every city around the world. But the problem is that a lot of studies that sometimes present urban data actually focuses on very distinct parts of the city, such as the urban park. And that's not necessarily reflective of the biological processes that are really occurring across the full gradient. So what you can see here is that from the most from the furthest from the city to the very city center, you can see that the different study sites have a different amount of impervious surface. Each data point is a nest box, and you can see the amount of impervious surface around every nest box that has been quantified in a radius of 100 meters. So I will walk you through some of these sites just so that you have an idea of how variable the environment is in a gradient of urbanization. So we are starting with the most outward study site. It's a suburban village and the natural forest. And so you can see that there is a lot of green in the natural forest. The amount of impervious surface is virtually zero because it is essentially a large protected forest just on the outskirts, 20 kilometers outside of Warsaw. And then if you go into the city, you can see that you have an intermix of different types of habitats. This is a residential area, while just on the south of this residential area, you will see a little urban woodland. You also have much larger swaths of green areas. And one such example is, for example, the Jewish cemetery, which is very much in the city center of Warsaw. Or you have the office area, which corresponds to the university campus. And you also have another residential area that we have been monitoring. So, sorry, maybe I haven't mentioned this, but every little black dot is a nest box that we have been following over the past years. And finally, we also have an urban park. So the typical urban site that people would normally do research in urban ecology. Now, as I said, impervious surface area is overall highly stable, highly repeatable in time, unless you are experiencing very intense urbanization. But there are also other important variables of the urban space that are much more labile. And the question is, well, to what extent these are actually repeatable? To what extent are they possible to exert repeatable impact on biological organisms? So in other words, some axes of the urban environment need to be repeatable in order to produce an impact on the wildlife. And so we have also demonstrated that, for example, human presence, even though it differs in space, it is a repeatable variable with a repeatability of around 0.6 if you include enough counts of humans on the site to essentially capture enough variation of human presence in the environment. Some variables are actually very poorly repeatable, and that is why it's important to be aware of this repeatability problem depending on the type of question that you are asking. And so just to give you an example, we have also been looking at the air pollution and the concentration of particulate matter in the environment. And you can see that depending on the measurement day, you will either have high pollution of particulate matter or low values of pollution. And similarly, air pollution can be highly variable in space. What you can see here is that you actually have some peaks of pollution that are likely to correspond to being household waste being burned by specific households, and that corresponds to this peak in particulate matter. So this has also been mirrored by some other researchers that have found that, for example, NOx pollution in an urbanization gradient is also poorly repeatable, and the time frame through which you measure different types of pollution in the air matters a lot for your inference. Now, if you actually plot the different environmental variables in a principal component analysis, you will see that these variables do covary with each other. And again, I'd like to focus on this impervious variable that is highly correlated with human presence, light pollution, and temperature, and even more so when we are looking at intra-city patterns. So, I have characterized the city, and the question is, well, what are the biological effects of the city that we can detect. So first of all, again, this is work from of Michela Corsini that has looked at growth curves and at the growth of passerine birds in the city. And what you can see here is that if we characterize nest boxes as being surrounded by either high ISA, so high impervious surface area in the vicinity, or low ISA, well, there is a weak indication that birds grow in a different trajectory in the low ISA environment. So if there is more green vegetation in the environment relative to those that grow in the high ISA environment. However, these trends are actually not significant from each other. Now, what's important to remember here is that in this particular data set, we only have birds that have survived throughout the entire growth trajectory. And that data set is completely different to a full data set of breeding events that we observe in the urban environment, because a lot of birds are essentially dying over time. And so if we look at that more closely, you will see that actually the effect of ISA, so impervious surface, on chick mass over time. So when birds hatch, there is no effect of ISA on the mass of chicks 2 days after hatching, but you can see that the further you grow, there is actually a highly significant negative effect of ISA on 5 days after hatching and 10 days after hatching. And similarly for blue tits, you see a similar pattern where essentially originally there is no effect of ISA on the mass of birds shortly after hatching, but then there is a very negative effect over time. And similarly for survival, essentially what we found is that there is a significant effect of impervious surface area in the close vicinity of nest boxes, especially at the level of medium and late survival stages. And it is possible that essentially we don't see the effect, any effect of ISA at the life history stage, at the late survival, just because most of the mortality has occurred in these intermediate life history stages. Now, another interesting analysis is related to the analysis of standardized selection differentials. So essentially what we can see here is that generally there is significant selection on the trait that we used. So that is mass after hatching, 2 days after hatching. And so it is true that there is a selection on mass for both species and whatever the environment we are looking at. However, what is particularly important here is that we can also see that selection on mass is significantly more, stronger in a high ISA environment. So in other words, if you hatch as a heavy bird, it matters much more for your fitness if you happen to hatch in a high ISA environment. So this trend is significant for gray tits, and it is not significant for blue tits, but you can also see that the point estimate of selection is considerably higher in a high ISA environment. So what can we say? So here there is a little bit of a preliminary data. Essentially, this year we have managed to finalize a 5-year-long data set of breeding data on the Warsaw gradient. And what you can see here is really, this is raw data, but what it shows you don't need any stats to really see with the naked eye that the impervious surface effect on the number of fledglings, on the number of individuals that are in the nest shortly before fledging out of the nest box, that there is a significant negative effect on the number of ISA, on the number of fledglings for both species, no matter which year you're looking at. And as I said, this is very much data in preparation, but if you just want to have a little bit of a ballpark figure and see, well, what is the impact of 50% ISA in your environment? How does that affect fitness? Well, we can really see that essentially fitness, here exemplified as the number of fledglings, that fitness is halved in both species with a 50% increase of ISA in the environment. So by concluding here about this part of the talk, you can see that we have found that impervious surface and the vicinity of your breeding event significantly has a significant pervasive negative effect on mass and fitness, and that a ballpark figure that we will need to refine in statistical models is that a 1% increase of imperviousness results in a 1% decrease in fitness loss. So, of course, what one may ask, well, okay, but why is ISA so detrimental to the fitness of birds? And as I was saying, ISA essentially exemplifies habitat loss, where essentially if you have concrete or asphalt, you do not have any natural life on that surface area. And one of the biological drivers of this decrease in fitness that should be investigated further is food. And this is a still of video analysis made by Michela Corsini that essentially shows what kind of food the birds bring to the nest in a gradient of urbanization. And if you were in a natural forest, you would essentially largely have caterpillars being brought to the nest and sometimes a little bit of spiders. But if you're in a city, you suddenly see that there is just this wide area of other types of food that are brought to the nest on a much more regular basis. And so if you think about food, so if you're thinking about how animals acclimate to the urban environment or adapt at a genetic level, but let's stay for the moment at this kind of phenotypic level, acclimation can be mediated also by essentially how you assimilate food in a radically novel environment. And one possible role into how animals cope is the role of the gut microbiota inside the gut of birds breeding, whether in an urban or rural environment. And so just to give you a little bit of a snapshot data, here there is some work that we have been doing with Anju Marachi and Barbara Kaspers at the University of Bielefeld. And I would like to present some data on the gut microbiota in the context of this reduction in mass caused by impervious surface area. And so we have looked at the changes in gut microbiota diversity in terms of alpha diversity or beta diversity. And what you can see is that there is essentially a lower species richness in the urban environment relative to what can be found in the rural environment. And also that impervious surface area specifically drives a shift in community composition of the gut microbiota in a gradient of urbanization. So the color coding perhaps is not ideal, but what you can see here is that essentially the further you go along this axis, the more you have highly urbanized sites, such as, sorry, I just don't see my legend. Yes, so the more you go, you have highly urbanized sites, such as the office area and residential areas in gray, so they're high ISA sites. And the less impervious surface you have, the more the microbiota samples cluster on this side of the graph. So by summarizing this part of the talk, essentially, I hope to have shown you that urbanization is essentially concomitant with environmental and biological stimuli that are outside of the natural ranges. And a ballpark figure of 1% imperviousness resulting in a 1% decrease in fitness loss will still need to be confirmed, but it is a ballpark figure that I would like to hold on to. And also we have found that there is stronger selection on mass at hatching in high ISA environments, and that essentially urbanization alters not only fitness and phenotype and fitness, but also internal gut microbiota. So all in all, there is a pervasive and negative impact of ISA on vertebrate passerine biology. Okay, so everything that I have shown you so far stems from data that has been collected in one city. And we know, as I have mentioned already in the introduction, that cities are everywhere, and the very exciting experimental framework that one might ask is, what is the potential of cities as essentially a globally replicated experiment of parallel evolution worldwide. And this is something that we wanted to address as well. And so, as I just mentioned, so far you have only seen data from Warsaw. But what if we have a glimpse of patterns that are occurring not only in one city, but in more cities across the country? And so essentially, we have performed an analysis of phenotype and genotype patterns across multiple cities, across 8 cities, where we have essentially caught birds that are just before the breeding season, so in February and March, across 8 cities. And this is work, this is tremendous work made by Arnaud da Silva, Marta Cele, Justyna Schulz, and Łukasz Wardecki. So essentially they have captured by MISNet territorial birds just before the breeding season in 2017 and 18. And again, we tried to really emphasize the need that not only do we want to look at these different cities, but also the misnetting occurred in 5 habitats in every city. So birds have been misnetted in natural forests. So these 4 large swaths of natural forest, suburban forests, natural corridors, urban parks, residential areas, and urban centers. And in total, 650 bluetis and greatis were captured throughout this initiative that lasted 2 years, just before the breeding season. And to answer the question about whether we see actually replicated processes. So this is the work of Marion Chatelain, who has done a lot of work on the dynamic of trace metal elements or heavy metals in biological tissues in a gradient of urbanization and in these replicated cities. And what you can see here is that essentially the PCA axis that captures the bulk of heavy metal variation in the feathers, you can see that essentially the closer you are to the city center, the greater the concentration of trace metal, heavy metal concentration in feathers. And what you have to remember here is that these values are essentially averaged over 8 cities. So there is a very clear trend that no matter what city you're in, there is a replicated trend for an increase in heavy metal pollution inside the feathers of the birds in these replicated cities, no matter and in both species. So another analysis that we went on to perform is to look at the genotypic properties of birds in these replicated urban system. And so we have looked at the Affymetrix. So we genotyped greatis using the Affymetrix SNP chip and to generate a high genotypic data set that could then be tested in the context of these different environmental variables. And so a first result that I would like to show is about the nucleotide diversity across sites. And what you can see is that essentially nucleotide diversity was pretty much equivalent across the 8 cities, the suburban forests, and the natural forests. However, if we actually then plug this allelic matrix into an RDA analysis, and we look at the first 2 axes of the RDA, we can see that the data points actually cluster very nicely based on city coordinates, so city latitude and longitude. So if you see, these are essentially the different cities that are included in the data set, and the green dots are essentially reflective of the large forest reserves that were also included in the analysis. So in other words, we know that there is genetic structuring in space. That in itself is not particularly novel. However, what was very nice to observe is that there is also a very convincing trend of urbanization here characterized by ISA at the sampling site, so at the place. So we derived essentially an amount of impervious surface in the vicinity of the misnetting site in the different environments. And you can see that there's a highly significant drive for an effect of ISA on genotypic variants when the entire data set is taken into account. So this is great, but then the question is, well, okay, maybe this trend is actually driven by 1 or 2 cities. And in other words, what we really want to know is whether there is a pattern for a change in genotypic frequencies driven by ISA in each of these particular cities. And so this is what I'm going to show you here. So not only were we curious to know, and yes, so I'm not sure if I mentioned that, but this work has been done by Sylwia Czarnomska, who did a postdoc in the lab. And so what we wanted to see is there an effect of ISA at the level of the city on the genotypic matrix? And also we need to remember that there is also this question of scale. If you imagine that we set up a mist net in a given habitat in a given city, how do we calculate what is the right scale for inference of the amount of ISA next to the sampling site? And so this is what we did here, is that what I'm showing you here is that not only did we vary are we here having results of a redundancy analysis of the effect of ISA on the allelic matrix in the different cities, but also when we take filter size of ISA that is of growing size around the sampling site. So what you can see here is that essentially there was a significant effect of, and highly significant in many instances, effect of ISA on genotypic variance in up to 4 cities out of the 8. There was an 8th city that I'm not representing here because we had a coordinate problem, so we need to rerun analysis for one of the cities. But what is quite striking is out of 7 cities presented here, and at a very small spatial scale and with a few numbers of a relatively small number of individuals, we see an impact of ISA on the allelic matrix for 4 cities. And you can also see that the amount that, depending on what spatial scale you're looking at, the signal of the effect of ISA on the genotypic variance will vary a little bit. So this is something that we need to explore further, but it essentially highlights again the fact that we really need to take into account a spatial scale when performing these analysis of urbanization on either biological or genotypic traits. So summarizing the talk a little bit, I hope to have convinced you that essentially we have convincingly shown that there is a radical change in the environment driven by the Anthropocene and driven by urbanization, whether we look at the level of Nestboxes, gradient of urbanization, or replicated cities. We have also detected a significant impact of urbanization on the phenotype, assessed as morphology, life history traits, or even trace metal and feathers. And we have also found an effect of urbanization at the genotypic level by looking at a SNP chip assay. And finally, there is also pervasive evidence that urbanization has an impact on individual survival with increasing amount of urbanization at the place where these birds are developing. So yes, and another thing as well that I haven't shown about so not only are we having these effects of urbanization on phenotype and genotype, but we can even dwell further and look at the extended phenotypes such as nests, and we know that there are differences in nest construction. And there is also an impact on the extended genotype that is presented by microbiota community assemblage in the avian gut. So if we look at this very nice graph that has been prepared by Charles Perrier et al. About the genomics of adaptation in the urban environment, it essentially summarizes what we would need to know to convincingly say that there is genetic adaptation in the urban environment. And so based on the data that we have collected in the past years, we can see that there are differences in phenotype. There are also differences in patterns of selection in the city, and there are also differences at the level of the DNA, and also differences at this level of the extended genome that is brought by microbes and microbiota in the gut. And what I think we should now move on to in the system is to essentially take more experimental approaches to look at candidate genes and to look at this microbiota in a little bit more detail. So these would be the further lines of inquiry that I think are worth pursuing in the future. So this is it for me. I think that I would like to finish by saying that when we're talking about adaptation to urban environment, there is also a liability, our liability as urban dwellers to play a role into how we view cities in the future, because there will be always a limit of adaptation, no matter at what level we are investigating it. So it is up to us really to decide whether we would like our cities to look like this, or whether we will just basically let it go and end up in cities that look a little bit more like from a Blade Runner movie. So with that in mind, I would like to thank you very much for your attention. And if the topic has caught your attention, a lot more is essentially discussed by some wonderful contributors in a book that has been recently out, that has been recently published, that I have edited together with Jason South and Anne Charmentier. And that will be it from me. One final very important slide, I would like to acknowledge a lot of people that have made this work possible. There has been a lot of very hard work from all the members of the Wild Urban Evolution and Ecology Lab that have collected data in the field and also analyzed it in the lab and in front of their computers over the past years. So I'd like to very warmly thank all these people that have generated data. And all the Spring field assistants that have worked very hard in the field, and also a lot of collaborators in Poland and in France and in Germany that have essentially made this work even better. And yes, so I would like to thank all of these people, because these results would not have been possible to generate without some great synergies between the different people listed on these slides. Thank you very much.
Okay, thanks a lot, Martha, for this great talk. So we have some questions on the Q&R. So one of them, or two of them, are by Gila. So Gila Ganem can turn on her microphone.
Yes.
Presumably. Gila?
Yes. So was.
Can you hear me?
Yes. Go
okay.
ahead.
Okay. Very, very nice talk and work.
Thank you.
I wondered, you know, the first part, the first act of your presentation, when you compared natural nest cavities and nest boxes, I wondered if the animals that would colonize nest boxes are those that are less successful in colonizing natural ones, and then it would mean that the differences in life history would be related to that, maybe.
I think it is largely unlikely, in the sense that there is natural cavities in this site or were in excess. And so essentially in the site where we followed nest boxes, there was also a lot of natural cavities. So essentially they could have gone into the natural cavities. And this is a little bit what happened in 2019, when great density in nest boxes was actually a little bit lower, and quite a portion of greates were then found in natural cavities in the nest box site. So that's data that I haven't shown, but it is unlikely that it is a question of being less successful, I think.
I'm not sure you got me completely, but I mean, I would expect the birds to be in natural cavity and in the nest box in the same place. But my question was whether there would be different individuals that would go for the nest boxes, because maybe it's more difficult, as you said, to colonize them.
Yeah, so I
like
okay.
Different personalities, or
yeah. Well, again, I would go. It's not impossible, but I would go back to the fact that the densities were highly equivalent between nestbox and natural cavity sites. So yes, I mean, personality-wise, these birds haven't been tested, so that's a fair point. I think that this is a limit of the setup that we had. Yeah, I guess this is something that could be looked further into.
Okay, thanks. Shall I ask my second question, Luis, or wait for the others?
Yes, you can go ahead. I think it's
okay. My question about the microbiota, I don't know anything about it, but I was surprised to see that the diversity of food that the animals have in town is higher, but the diversity of microbiota is lower. So I wondered if it is something that is expected generally, or it's a surprise.
So that's a very good comment. Essentially, actually, reports in the literature are mixed about alpha diversity. So this essentially microbiota richness in the city. Some studies really do report that there is actually higher alpha diversity in cities. And it's important. So yes, so essentially, this is very preliminary data, and it definitely needs to be investigated further, because there is also not just the question of species richness, but also the type of ecosystem properties of these different types of bacteria. So that's essentially a first part of an investigation that that needs to be looked into with more detail.
Thank you.
Junk food in the cities, there's a comment. Okay, so Mathieu Joron, who likes predation a lot, has a question about predation. Someone can turn on his microphone.
Hi, Marta. Can you hear me?
Yes, I can.
Yeah. Okay. Nice talk. I was wondering. We often hear that living close to humans makes the habitat devoid of predators. Maybe that doesn't really match the case of nesting birds because of cats. But I was wondering if there was any correlation between a predation gradient and the ISA gradient, or is there a, how does predation vary with center to distance to city center, or with ISA?
So essentially what we're really lacking is a longitudinal follow-up of bird movement after they leave the nest. And that's something that would answer a lot of questions that for the moment are unanswered. But the thing is that a lot of the predation that is occurring in natural environments often occurs, well, we at least know that a lot is occurring during tit development in the nest box, such as woodpeckers. There's definitely fewer of them in the city, so overall predation rates are higher in the natural environment during chick development. But we don't really know what is happening later on. So I do agree that definitely there are more cats in the city. And I wouldn't be surprised if it is to some level correlated with the amount of impervious surface. The question is that it doesn't really necessarily implies that there are fewer predators later on in the natural habitat. So that would just need more work, and we don't have any precise quantitative estimates about them. So I think it's quite difficult to speculate on that.
Okay, thank you.
Okay, there's a question by Zun Mao. Do you want to turn on your microphone? Yes, no, person left apparently.
So I see the question. I'll just answer it straight away. Essentially, the nest boxes and natural habitats were not really geographically separated. They were separated by 200 meters, and both stands do not differ in light pollution, noise pollution, air pollution, and also most importantly, perhaps geographical, phytosociological composition. So essentially, it's the same forest.
All right. Sorry, I'm going to skip Yanis for now.
Okay.
I like doing that because it seems like the Philippa Guillerme's question is related to the previous one, in my understanding. So Philippa can turn on her microphone if she wants to have
Yeah, okay, cool. So how would you deal with areas with high ISA but with high vegetation cover, green roofs or tree cover in paved areas? So essentially what I have shown in the principal component analysis is that we have a very, very clear negative correlation between ISA and tree cover. So at least in Warsaw, green roofs are not yet in, but I think it would be a very nice question, you know, when green roofs, I mean, to be entirely honest, I just don't think that the green roofs will gain the kind of huge momentum that would be required to make a real impact onto this negative correlation between tree cover and ISA. But, and actually even I think in those countries, including France, where these regulations are necessary, governments are caving in for example, also allowing solar panels and roofs, because it's just very, very difficult to have green roofs. Mm-hmm. So, but overall, yeah, so like as a ballpark figure, there is definitely a strong negative correlation between both. So I think that these kind of situations where we would have high ISA and high tree cover, well, essentially, I guess it would be just a better environment than in an environment where you have high ISA and low tree cover. But at the end of the day, this is something that you then essentially deal with in a model selection framework, where you essentially can where you basically decide on which variables you fit in a model. So I think this is something that just can be statistically addressed, specifically if you essentially formulate your biological hypothesis, and then you go from there.
Okay, so Philippa seems to have left, so she can see the answer on YouTube, I guess. So yes, Yanis Michalakis had a question having to do with local adaptation. So perhaps we can turn on his microphone, or he can turn it on. I don't know.
Am I really allowed to ask it?
Yes, you can.
Thank you. Thank you. So thank you for the seminar. I had two questions, but I guess you won't be able to answer them from the answers you gave to the other ones. Anyway, is there any indication that these birds may adapt to the urban environments? For instance, do the ones that live in high ISA environments do better than birds from the woods, or vice versa? That's the first question. And the second one is a bit related, and actually related as far as I can tell to one of Gira's questions, which is whether there is an indication of habitat nest choice. That is there an indication that birds that are born in high ISA nests tend to settle more, breed more often in high ISA nests than the contrary?
Yeah, so these are all very good questions, especially in an evolutionary perspective of adaptation to urban environment. And I'm sorry to say that the data for that is at the moment is still very scant, just because birds as a study system are great for some questions, but less so if we're talking about recruitment and experiments in the city. So your question essentially has been answered in simpler systems such as ants. We have, so Sarah Diamond has done some great work where she essentially where she essentially reared rural ants in the urban environment and urban ants in the rural environment. And essentially she showed that that ants are adapted to their original environment, and that rural ants reproduced more poorly in the urban environment and vice versa. So in other words, she demonstrated based on the kind of based on a thermoregulation argument and a heat island argument that these kind of things can be observed in some organisms. In terms of birds, I don't have this kind of data, but definitely one powerful approach is to essentially use cross-fostering, and that is something that I would like to implement in the near future. But I don't have any cross-fostering data to discuss just yet.
Thank you very much. Can Louis allow me a third one?
Yeah, go ahead if you want. Yeah,
so after what you said in the beginning, should I remove the tit nest boxes I have in my garden? Am I breeding crooked tits?
Where would you like to move them?
I'll just take them away.
Why?
Well, if I'm
Well,
helping, if I'm growing tits that are maladapted to the natural environment, perhaps I should stop. I don't know.
Well, if you're living in a city, my argument would be is that essentially, this is actually something that gave me comfort when I was setting up the Nestboxes initially. I thought like, well, even if we won't be able to get any data out of it, at least there are Nestboxes in the city. This is the cool thing about, from a conservation point of view, is that, you know, essentially I started with discussing natural cavities, but the thing is that in the city, most trees don't have any natural cavities. So if we do want to have wildlife in the city, you really need to provide nest boxes. And so this is an opportunity to essentially generate wildlife in the city. So no, I wouldn't remove your nest boxes from your garden or balcony.
Well, I'm in the countryside, actually. Thank you anyway.
What's your ISA, Yanis? You need to measure this.
It must be very low. Martha should come measure it.
Just I had a quick question about also relating to local adaptation. Did you look at what parts or the parts of the genome that were correlated with ISA in your RDA analysis? Did you find anything that seems interesting in there, or you didn't really check?
Yeah, so that's essentially data that is ongoing. So that's definitely on the cards, and we are running some analysis right now. But we don't have any results of that. So of course, the idea would be to look at essentially genomic outliers by performing different types of genomic analysis, and to see whether there are some overlapping genomic outliers depending on the method. But we haven't gone down that route just yet. I mean, we do have some first selection of genome areas of interest by one analysis, but we just want to validate it with some others. So I don't have any data for the moment to share about that.
Okay, so ongoing. Okay, and last question will be by Gauthier Daubigny. So I'll just, you can turn on your microphone now, I think. Or
you hear me?
Yes.
Yes. Thanks for this very nice talk, Marta. My question is about a bit similar than the one asked by Mathieu Geron. He was talking about predators. What about pathogens? Could it be that pathogens diversity and abundance could explain at least a good part of the observed differences between your different sites?
Yes, so that's again, this is a very good question, and we've only started looking into that. So there is a researcher, Dr. Eva Mijajewska, who is actually looking currently at incidence of avian malaria in the multi-city data set. So we'll be having some data shortly. We also know that there were different incidents of ticks in the city, and particularly there were more ticks in natural cavities, in birds breeding in natural cavities than birds breeding in nest boxes. So again, you could also claim that there may be some different patterns of disease transmission depending on whether you breed in a natural cavity or in a nest box, but the sample sizes, unfortunately, here are quite small. Yes, so that's essentially to some level work in progress. And also Irena Dilice will be looking at incidence of avian malaria between natural cavities and nest boxes as well. So again, this is work in progress.
Cool, thank you.
Thank you.
Okay, great. So let's just thank Marta again. So just to reiterate that she's here all year and she speaks perfectly French for those who prefer speaking French. And okay, so just to tell you that the next talk next week, the next talk of the year will be by Clara Torres Barcelos, so also another person who was in Montpellier before and now has a position at INRA in PACA. So see you next week.
Thank you very much. Thank you.